Hi, my name is Christine and my presentation is on the topic of mechanobiology. So before I begin, I would like to give a little review of the cell. Cells are the basic unit of life. They make up all living things and are living organisms themselves. They have many basic functions such as giving shape and protection to our bodies and absorbing nutrients to convert it into energy we can use. They also have specialized functions that make them more distinct from one another, which can be seen in our muscles, brain, skin, or blood cells. Higher level of cognitive thinking, memory formation, and feelings of complex emotions such as love or self-sacrifice are also due to the specialized cells in our brains, also known as neurons. Although they are the most basic unit of life and seem very simple, together they can create increasingly complex and intricate organisms. The reason why they can create such complex organisms such as ourselves is through communication and working in unison. Communication is crucial for creating and maintaining a healthy organism. We are most familiar with the chemical mechanisms of cell communication, which are through things such as hormones or neurotransmitters. But we also know that cell-to-cell contact is also an important way of communication since they are physically touching each other. Since we study cells on such a molecular level as individual organisms, I often forget that they work together in some way to create a whole organism. As I mentioned before, cells can communicate chemically, which usually involves cells that are not physically touching. These cells can either be really close to each other or even far apart and communicate by sending chemical messengers through the blood. They can also communicate physically via mechanical forces. Cells can detect and respond to mechanical stimuli that result from physical changes in their external environment. This involves the process of mechanotransduction, which includes any mechanisms by which cells convert mechanical stimuli into electrochemical activities to activate internal signaling pathways. So what are some things involved in mechanotransduction? Cells have mechanosensory proteins that can detect physical changes in the environment, which can cause a cell to modify their physical properties in response to this change. It usually involves the stiffening, softening, or reorientation of the cytoskeletal filaments. The figure on the top right shows that different proteins react to specific stimuli. For example, some will respond to heavy pressures, while others will respond to a stretching force. By changing the shape of their cytoskeletal filaments, it can cause ion channels to open or close that can lead to changes in the cell's chemical composition that can cause a downstream reaction of various signaling pathways, also known as signal transduction. Signal transduction is the most common method of cell communication that will lead to a response. The bottom figure on the right shows a simplified version of signal transduction, which starts and begins when cells get a stimulus from the external environment, whether it is a chemical messenger or a mechanical stimuli. This can lead to various signaling pathways within the cell that will eventually lead to a response, such as gene expression. One of the most important structures involved in mechanotransduction is the ECM, which is the extracellular matrix. The extracellular matrix is the space surrounding the cells that make up the non-living portion of the tissue. It is composed of various proteins that keep all the cells connected into one network or organ. It functions in detecting changes in the physical environment and relaying it to the individual cells that it encompasses. It is thought to be one of the first steps initiating mechanotransduction. It also influences many cellular functions such as migration, differentiation, growth, and survival. Integrins are transmembrane proteins and mechanosensory proteins on the cell that connect the ECM directly to it. Other mediators involved in mechanotransduction include the cytoskeleton and other proteins connecting cells to other cells. It also includes the nucleus, which elicits the response, such as expressing or inhibiting certain genes and proteins, or by changing the cell behavior. So what exactly is mechanobiology? Mechanobiology is a relatively new field of study that uses biology and engineering to figure out how physical forces and changes in the mechanical properties of the cells and tissues affect cell behavior, such as their development, differentiation, and abnormalities that may lead to disease. Although it is still relatively new, the idea that physical forces had an effect on cell behavior is not a new concept. It goes all the way back to 1892, when Julius Wolff, a surgeon and anatomist, published a book in which he shows that the bone structure adapts permanently to the loading conditions, like how much a load a person endures daily, which can differ from someone from a construction worker versus an office worker. He even regarded his work to be the brick to complete the building of Charles Darwin. His work also established orthopedics, which specializes in bone and muscle as an independent discipline. The reason why mechanobiology is still an emerging field is because for most of the 20th century, scientists were focused on molecular biology and analyzing the genetic basis of life. It was also due to the lack of advanced technology. But now that technology has improved, we can finally start to understand how cells can take physical mechanical forces and convert it into chemical signals. The processes of mechanotransduction can be studied better to show the roles that mechanical forces have in regulating cell behavior. Mechanobiology tries to understand the interactions between the cell and its environment, how they are able to detect, measure, and respond to physical forces to maintain homeostasis. Cells respond to these forces and adapt by changing their cytoskeleton shape and properties. There are three general models in mechanobiology that explains how cells regulate their cytoskeletal network when they are responding to a mechanical stimuli. The first one is the tensegrity model, which states that cells respond to a load or force by using the pre-existing tensions within their cytoskeletal fibers, where they will push or pull the fibers against each other to match the strength of the force applied. The semi-flexible chain model states that the fibers act like semi-flexible chains that are uniformly distributed throughout the cell, which will stiffen or loosen in response to a load. These fibers will act in unison, so the entire cell will stiffen regardless of where the force is applied. The last model is the dipole polarization model. It's similar to the semi-flexible chain model because the fibers are uniformly distributed throughout the cell. However, the direction of where the force comes from is taken into account. Although the fibers are still uniformly distributed, they don't act in unison. The fibers form dipoles with each other, causing the fibers to pull on each other due to the polarization. The polarization propagates throughout the cell depending on the direction of the force applied. What these three models have in common is that the mechanical stimuli must pass through the ECM first before reaching the cells. The ECM supports and transmits the loads to the cells. The fibers in the ECM also provide a mechanical memory to set up a baseline for what homeostasis should be. So if we put it all together, when there is a mechanical stimuli or force applied, it travels through the ECM first. The ECM will then pass the stimuli to the mechanosensory proteins located on the cell membrane, which will induce a change in the cytoskeletal network by either changing the shape or property of the cell. This change can also induce a signaling transduction pathway by opening ion channels. The signal transduction pathway can change the cell's behavior by either expressing certain genes or proteins, signal the cell for differentiation or growth, or even program cell death. Basically, mechanical forces can cause the same chain of events as chemical signals. Now that mechanotransduction can be studied in more detail due to technological advances, studies are showing that mechanobiology diseases are a lot more common than previously thought. These diseases include any disease that is caused by a disruption in the cell's mechanotransduction processes, mainly between the ECM, cytoskeleton, and nucleus. Almost all cells rely on mechanotransduction signaling for normal function, so you can see how wide mechanobiology diseases range. This figure was taken from a research article by Diana Jaluk and Jen Lamerdine, which generalizes the ways that lead to a mechanobiology disease. There can be changes in the extracellular environment, such as deformations in the tissue or changes in the cell's physical property like mechanosensory protein composition or changes in the composition of the ECM. There can also be changes in the cell structure and organization, which usually involves mutations or abnormal functions in the proteins rather than changes in the composition. There can also be changes in cellular sensing and signaling, which involves disruptions in the proteins involved with the signaling pathway. These all lead to some disruption of mechanotransduction signaling, which may lead to a disease. This table was taken from a review article by Donald Ingber, which lists a variety of mechanobiology diseases including heart failure, cancer, migraines, stroke, stuttering, asthma, and even sexual dysfunction. In his article, he states that the current focus of medicine tends to focus on the molecular and chemical genetics behind diseases and disregards the physical aspect. However, many diseases cause or are caused by changes in tissue structure or the mechanics of cellular processes such as mechanotransduction. But it is not to say that the chemical or molecular aspects are less important. Rather, they all have a significant role and interact with one another to affect cell behavior. So how do disruptions in mechanotransduction lead to disease? The same research article by Jaluk and Lamerding shows how disruptions in these processes and the ability of cells to respond to mechanical stress could be implicated in all types of diseases. They highlight the importance of cells to be able to respond to their physical environment because it is critical for the development and maintenance of tissues exposed to mechanical stress, such as muscle or bone tissue, as well as tissues in the lungs, immune system, and nervous system. It is also crucial for physiological processes that affect the organism as a whole, such as blood pressure and blood flow. Their research paper uses cardiac muscle cells as an example of how defects in the mechanotransduction processes can lead to heart disease. Heart cells or cardiac myocytes are mechanosensory cells that can directly respond to mechanical loads and stresses through internal mechanosensors, such as stretch-sensitive ion channels, integrins, and other receptors. These mechanosensors activate many signaling pathways, which in turn trigger the expression of genes that cause the heart cells to increase in width or length. This allows the heart to sustain prolonged stress. The increase in cell size due to mechanical stress is known as a hypertrophic response, which has two categories. It can be a physiological hypertrophic response, which results from exercise or pregnancy that causes changes to the cell size in proportion to the mechanical stress. It can also be a pathological hypertrophic response, which results from abnormal changes in the cardiac workload, such as hypertension or heart attacks, that causes the cells to change sizes disproportionately. This can cause structural changes of the myocytes and ECM that can lead to apoptosis and necrosis, eventually leading to heart failure. The exact mechanisms that govern which response the heart will undergo is unknown. They also show its role in cancer. As I stated before, mechanotransduction and the ECM have a critical role in cellular development and differentiation. For example, during early embryonic development, cells are programmed into left-right patterning when developing into organs by cilia-driven leftward flow of fluids during gestation, and mechanotransduction signaling that induce expression of growth factors on the left side of the embryo. Cartagener syndrome is a rare genetic disease that disrupts these processes and results in left-right reversal of organ development, which causes abnormal placement of organs, frequent respiratory distress, chronic nasal congestions and ear infections, or hearing loss. Cartagener syndrome is an example of how crucial mechanotransduction is in cellular differentiation and development. The ECM, cytoskeletal tension, and mechanotransduction signaling pathways are important factors that can contribute to cellular malignant transformation, production of tumors, and metastasis, which is the spreading of cancer. So exactly how can these factors contribute to cancer? Increased stiffness in the ECM, which can be caused by the thickening and scarring of connective tissue, can lead to changes in the cytoskeletal tension. The increased tension and stiffness are sensed by the integrins on the cell that connect the ECM with the cytoskeleton, which activate a signaling pathway known as the Rho-ROCK pathway. The Rho-ROCK pathway increases the cytoskeletal tension even more, which can also further stiffen the ECM, creating a positive feedback loop in which the tensions and stiffness of the cell keep increasing. The increased tension and stiffening of the cell and the cell's environment can change their behavior and override their normal functioning genes to turn them into cancerous cells that keep dividing uncontrollably. This can be shown by the difference in the physical properties of a cancerous tissue versus a normal tissue. Tumors are generally much stiffer than the surrounding normal tissue. As you can see, the conditions of the cell's external environment has a huge impact on dictating cellular processes. Even if the cell is functioning properly, the external environment can cause a healthy cell to defect. For example, atherosclerosis is the narrowing of arteries due to plaque buildup that can lead to heart attacks or strokes. This can be caused by disturbances in fluid stress caused by the plaque obstructing blood flow that lead to vascular remodeling, meaning that the cells will change its properties in response to the change in its external environment. Furthermore, a study showed that mice with dehydrated brain tissues had impaired neuronal function. In order for the neuronal cells to function properly, they must be in an optimal hydrated environment. In order to show the relationship between the cell and its external environment, researchers placed 3 different types of human brain cells into a culture with fully hydrated 3D collagen gel to act as the ECM. They altered the culture's water activity and found that over time, the cells contracted the gel and expelled fluid in proportion to pressures applied, meaning that they changed their contractive forces to adjust to the environment and prevent excessive dehydration and hydration. However, if the external change is too great, in which the cells cannot adjust properly, it can lead to disruptions in cellular processes because the cell cannot handle the excessive load. Now that mechanobiology is recognized in a number of diseases, perhaps they can use this to fill in the missing links. Although healthcare and technology has advanced greatly over the past few decades, leading to increased life expectancy and well-being of people, there are still many unknowns in the realm of diseases. Now that technology has given us the ability to study cells in great depth, we can see that there are multiple factors that contribute to disease that relate to one another and influence one another. Previously, the chemical and genetic aspects of diseases were focused upon, but now a whole new side of mechanical and physical aspects can be studied and how it intertwines with previous knowledge of molecular genetics and chemical processes. The study of mechanobiology can also provide a wider range of treatment options as well as new ones. Mechanotherapeutics is a field of prevention and treatment of diseases that utilizes the concepts of mechanobiology to develop new types of drugs that target the mechanotransduction processes, as well as devices that can be worn by the patient to leverage physical strengths. There is also a field called tissue engineering, which recreates living tissues using biomaterials and biologically active molecules to restore function to organs, or even producing whole functioning organs that can be transplanted in an organism to replace lost or functioning organs. Right now, it is mainly being used for research in drug development rather than actual people, but I believe in time it can be used to help those on the organ transplant waiting list. I believe that mechanobiology has great potential to change current views and methods in research and clinical applications for the benefit of the human population. This field can provide the missing links in unknown aspects of diseases and improve treatments. I also think that it can broaden perspectives to allow us to consider using other specialized fields such as math or physics to further uncover unknown aspects about the biological processes that give us life.